93% of US grown corn is on the high-risk status. And as for every other crop in America, the medium-risk status. Apparently, there's no scientific consensus about these things that we call GMOs. So you read a headline like this and you think to yourself, is that actually true? Because if that's true, how is it legal that a company could be pushing something so dangerous as GMOs, as everyone would make you think they are, without any repercussions? So, are GMOs really as bad as people say? Well, let's talk about the science behind that.
Welcome to The Science Behind That with Atticus Hamilton.
Hello, all you scientists. Welcome on back to the Science Behind That podcast. I am your host, Atticus Hamilton, and on today's episode of The Science Behind That, as you heard in the intro, we're talking about apparently a controversial subject, GMOs. And so this is going to be a fun episode today. I think that it will be actually a pretty short episode because the reality is that there's not that much information to cover. It's a pretty quick sort of thing. So, but before we get into this, ladies and gentlemen, please go grab yourselves a nice hot cup of coffee, add a little bit of maple syrup to it, and let's jump right into the science behind that. So, today we're talking about GMOs, and apparently this is a really controversial topic. And personally, I've never understood why it's a controversial topic, and I hope that, by the end of the episode, I can convince you of that as well. Now, with topics like these that are loaded with all this sort of controversy, I always like to put out here too, my purpose of doing this episode and every other episode is just to give you guys actual science and to talk about the real science behind these things, the science behind everyday life. I have done episodes in the past, ones on testosterone, where I've gotten very angry comments by people who didn't like it. And, that may happen again. But ladies and gentlemen, the reality is we're talking about the science, no politics, no opinions, just the facts. And the fact is, short summary, fact is, there is no literature that suggests that GMOs have any direct cause for long-term health effects of consumption, assuming we're looking at the GMOs that were created for human consumption, right? I mean, if you eat a GMO that's not designed for human consumption and you get sick from it, well, that's your fault. That's not because of the genetically modified organism. That's because you ate something that you weren't supposed to eat, right? And we're going to get into this. So when I was doing research for this episode, I came across this funny website called the non-GMOproject.org. And it's very interesting because obviously based on their name, you can assume that they're against GMOs, right? And they have all these different sub-pages to their website here. GMO facts, what is a GMO, GMO science, understanding risk status. For GMO facts, it was kind of funny. They say there's no scientific consensus on the safety of GMOs. According to a 2015 statement signed by 300 scientists, physicians, and scholars, the claim of a scientific consensus on GMOs frequently repeated in the media is, quote, an artificial construct that has been falsely perpetuated. They continue to say, to date, there have been no epidemiological studies investigating the potential effects of GMO food on human health. But then they say most of the research that is used to claim that GMOs are safe has been performed by biotech companies. So there's a lot to unpack here. And some of you may be like, Atticus, why are you talking about something that goes against, what your facts lay out? And the reason is because it's important to examine both sides of an argument like this, especially when there's, a massive amount of controversy. The reality is that this statement that they make here, according to date, there have been no epidemiological studies investigating the potential effects of GMO food on human health, is fallacious. There have been studies. I pulled up one here. This was a study titled Human Health Concerns with GMO Crops, published in the Journal of Mutation Research and Reviews in Mutation Research, November 2003, volume 544, issues 2 through 3. And ultimately, this research investigated all of the literature that is out there. And I understand that this is an older paper, 2003, but it examined all the literature, all the research that is out there from 1990 up to 2003. And ultimately, their consensus was that because people have this perceived concern about GMOs, they're one of the most highly regulated areas of the food industry. And not highly regulated in terms of, saying what you can and can't put on the packaging, but in terms of safety standards. And this paper goes to say that the scientific evidence indicates that biotechnology-derived foods are as safe and nutritious as conventional counterparts. Using established and accepted methods of analytical, nutritional, and toxicological research and the concept of substantial equivalence, the safety of current biotechnology-derived foods can be compared with that of their conventional counterparts. And, to me, I always like meta-reviews. And I like meta-reviews for one massive reason, which is the purpose of a meta-review is to look for all the research that has been already published, right, of a specific topic, and talk about that research and basically summarize the overall claims. And that is a really nice way of doing things and of getting information. And this was a meta-review, and it was a meta-review of all of the, at the time, present research in the area of toxicology, nutritional health sciences, etc., and they determined that there's not any literature that suggests that GMOs are bad for you. Now, for those of you listening, if you guys have any research papers or whatnot that you'd like me to take a look at, you can always send them to my email at thesciencebt@gmail.com, and I'd love to take a look at them. But so now we have this foundational framework set up. We've talked about what the one side that's very anti-GMO is saying. We've talked about the other side that is, I guess, pro-GMO and what they're saying. But we have to get down to the research. Sorry, I had to take a sip of my coffee. So it's kind of interesting. So this website, again, that I mentioned before, they have for a comprehensive overview of available research on GMOs, please download the report GMO Myths and Truths. And so this is from a place called Earth Open Source. And the interesting thing here is that this organization, I guess, is geared towards developing farming and agriculture away from GMOs. So grassroots agricultural development, which I'm down for small family farms. I think that's a lot better than factory farms. However, the reality is that I think that this website that you're directed to here has a very obvious bias against GMOs, and the irony here is, right, if you're looking at a piece of research from, a biotech company that says, hey, this product we've created is fantastic, you wouldn't trust that as much as you would from an independent source, right? Because that has a bias. So this should be the exact same way. So anyway, let's talk about the science. What is a GMO? Well, in a most basic sense, a GMO, which stands for genetically modified organism, is an organism that has had its genome altered in some way, shape, or form. And in this episode, we're going to talk about two main examples here that I'd like to highlight this idea, or why the idea that GMOs are fundamentally bad is wrong. And those examples are corn and insulin. So starting off with corn. So a long time ago, I did an episode about botany. I think it was about like the evolution of spice and why we like spicy foods. And that episode, I dealt a lot with hunter-gatherers and the progression from a hunter-gatherer society to an agricultural-based society, and this concept called selective breeding. And so what is selective breeding? Selective breeding is where you take an organism. It could be a plant like corn, it could be a cow or a sheep or whatever, and you take individuals from a group of those organisms that possess a desirable genetic trait, or phenotypic trait, I guess, and you breed those individuals only in the hopes that you not only perpetuate that gene that's causing that phenotypic expression, but that you also get a more and more exaggerated phenotypic expression of whatever that gene is. In basic biology, we oftentimes refer to this as Mendelian genetics, right, with Gregor Mendel and his peas. And for those of you who don't know what I'm talking about, Mendel was a monk, and he's kind of considered the father of modern genetics because he basically discovered the idea that, phenotypic traits in the case of his beans of white flowers or blue flowers or a wrinkled, shriveled pea and a big pea is inheritable. And so that's where the Punnett square comes from, and that's where this idea of dominant and recessive traits comes from. Now, I could do a whole episode on the history of genealogy, basically, but that's for another episode. But ultimately, in his research, not only did he tell us about genetics, and not only did he discover genetics in a way, but it also tells us something interesting, which is selective breeding, right? Because he learned that if he took, let's say a pea that maybe only had 3 peas in the pod, but they were massive, and only bred that pea plant with other pea plants, those peas would progressively get bigger and bigger and bigger. And it's kind of fun. You can try that at home. I've done that experiment at home. And after around 3 generations, you'll end up with peas that are like one massive pea inside of a pea pod, right? Now, we don't consider that GMO, right? Because we're not directly altering the genome, but that's selective breeding. And so in a way, that is a type of genetic modification. And so how does this get back to corn? Modern corn comes from a weedy grass that existed around 10,000 years ago called teoscentate, and that grew in Mexico. And go to Google, look this up. This is so cool. Teoscentate is Teosinte. I'm sorry if I'm butchering the pronunciation of that name. But it looks very similar to like wheatgrass. It had these corn kernels, kind of like what we see today, but they were encased in a very hard silica shell, pretty gross to eat. But what happened was our ancestors around 10,000 years ago started selectively breeding for strains of teoscentate that had larger and larger kernels, basically, until, here we are 10,000 years later, and we have these massive ears of corn that don't exist in nature. They are completely developed by humans. So, but we don't consider those GMOs, right? Because we didn't directly go into the genome of that plant using CRISPR or a viral delivery system and remove a gene and insert another gene. But we did modify the genome of teosinte to give us modern maize. It just took 10,000 years of selective breeding. Maybe a little less. But it took a very long time, and that's all selective breeding is. Selective breeding is where you take a group of organisms and you select for a phenotypic expression of a genotypic trait, right? And over time, you get a more and more accentuated expression of that gene. And in the case of corn, it was to go from a nasty little silica-like kernel pod thing that I can't imagine how gross that would be to eat, to a modern ear of corn that I think a lot of people around the world love. Millions, probably billions of people eat corn on a daily basis. And so an argument I see a lot of people make about why GMOs are bad is because they're not natural. And but then they'll eat corn, right? And so the fact is that corn isn't really natural either. Modern maize isn't natural either. That has been genetically altered through a very long process called selective breeding over a very long period of time, but essentially it's not that different from genetically modifying an organism, right? It's just it takes a lot longer. Now, how does that lead us into GMOs? Well, we're going to get to that, and I'm going to explain that through the example of modern insulin production. But before we do that, let me just talk about briefly some ways that we can genetically modify organisms. So an example of a way we can do this is the CRISPR-Cas system. Now, there's a lot of biochemistry and genetics here that I'm going to kind of try my best to simplify, but essentially CRISPR-Cas9, and there's a lot of different CRISPR-Cases. There's like CRISPR-Cas9, 10, 11, and 12. But it just refers to an enzyme called Cas. But anyway, this is just essentially a mechanism where we can take a segment of genome and we can cleave out a specific gene and we can replace it with another gene. Glowfish are genetically modified organisms. That's right. The glowfish that you see in Petco are genetically modified organisms. They were, in fact, genetically modified using CRISPR. And what was done was we took zebrafish. Generally, now you have like tetras, I think, are glowfish now. But originally it was just zebrafish. But they take zebra fish, and in the embryo of the fish, before it's born, basically, you use CRISPR, and what researchers did was they inserted a gene from jellyfish that was a biofluorescent gene. And so they used CRISPR to insert that into a non-coding region of the genome to make it a coding region of the genome. And they inserted that gene into the genome of the zebrafish, and that gene encodes for a protein that makes the fish biofluorescent. And then the embryo that was born, it became biofluorescent. And in fact, researchers have not only done this with fish, but we've done this with mice as well, and hamsters and rats. So yes, you can Google it. There are biofluorescent mice and hamsters and rats out there. These are genetically modified organisms. And if you were to eat them, I mean, I don't know why you would, but if you were to eat them, they wouldn't make you sick. And if you did get sick, it wouldn't be from the genetic modification. And why am I so confident in saying that? People eat jellyfish all the time, and the gene came from jellyfish. And all the gene that was inserted into glowfish is doing is it's just allowing the fish's cells to translate a protein that is just a fluorescent protein, right? So it fluoresces. And that's all it is. And so why do I bring this up? I bring this up because that's all a genetic modified organism is. Now, the way you can go about genetic modification is different. So you don't have to use CRISPR. There are examples of people using viral delivery systems to modify organisms. Why viruses? Because viruses are fantastic at getting into cells and inserting their DNA into cells and reprogramming the cell, essentially. And for some organisms, you don't even need that. For some organisms, all you need to do is insert a plasmid into the environment that organism is in. It will absorb that plasmid, and it will start to transcribe whatever gene that is, whatever gene is encoded on this plasmid. And this leads me to the final part of this, which is insulin. So once upon a time, insulin was produced almost exclusively from pigs, from pigs' pancreatic cells in the laboratory. And that's why it was very expensive. And it still is very expensive, but it's no longer produced in this way. Now, originally, you would produce these insulin from basically pigs' pancreases, but it would take a lot of pigs, it would take a lot of pancreatic cells, and it would take a very long time to produce this, right? Now, this gets us into plasmid genetics. I'm going to spare you a lot of details, even though I do really love this. I took a class in college called the Molecular Genetics of Bacteria, and we talked about plasmids and plasmid genetics, and it is so cool, but unfortunately, I'm already at 21 minutes and 15 seconds here, and if we went into plasmid genetics, this would be like a 4-hour episode, so I'm not going to. But ultimately, for the most part, only bacteria and archaea are able to absorb plasmids. Now, what are plasmids? Plasmids, it's basically just a circle of genetic material. And a lot of times that circle of genetic material will just contain a gene and then either an RNA or a DNA polymerase attached to it. And so in that case, it's called a self-replicating plasmid. And most of the time, it's just prokaryotes that are able to utilize plasmids because prokaryotes have one large circular chromosome as their genome. Whereas most eukaryotic cells, or actually all eukaryotic cells, have linear genomes. However, yeast are eukaryotes, but they are able to utilize plasmids, and they're able to uptake plasmids, which makes yeast and other fungi very, very, very unique in terms of the eukaryotic world. So what researchers basically discovered a while ago in the early 2000s is that if you take pig pancreatic cells and you use CRISPR to remove the gene that encodes the production of insulin, which is just one gene, you can insert that gene on a plasmid and you can put that plasmid in a petri dish, basically, or a suspension that has yeast in it, brewer's yeast, actually, that's used for the fermentation of beer. If you put that plasmid with that gene for insulin on it in a test tube environment, essentially, with yeast cells, what's going to happen? The yeast assimilate that plasmid, and because the plasmid is self-replicating, once they assimilate that plasmid into their bodies, even though it's not a body, it's just a cell, into their cellular body, they start producing insulin. And they start producing a lot of insulin because to the yeast cell, this is a protein that is neither necessary for survival nor harmful for survival. And so because yeasts use plasmids already to transfer information back between each other, there's no reason to inhibit both the replication of this plasmid and the translation of the insulin protein on this plasmid, right? And so the yeast just start translating a ton of insulin. And when I say translating, what I mean is transcription, right, where you take the DNA, you produce a strand of mRNA off of that plasmid. That mRNA then goes through the ribosome, which translates into a protein. But anyway, so it just starts translating a bunch of insulin. And researchers discovered, oh my God, we've genetically modified brewer's yeast to produce insulin that we can now use in the pharmaceutical world for patients with diabetes. Because it's cheap, it's very easy to do, and it's safe. Let me restate that again. It's cheap, it's easy to do, and it's safe. And these are GMOs. So for all of you listening out there with either type 1 or type 2 diabetes that take insulin on a regular basis, your insulin came from genetically modified yeast. But it's keeping you alive. Why? Because there's nothing wrong with it. It's a marvel of modern science. It's something that if you would have told me we were going to do in 1980, I would have said you're crazy. It's a marvel of modern science, and GMOs for dietary consumption are no different. There's no literature from credible sources to suggest that GMOs have any negative effect on human health. And if you guys know of one, I would love to read it. And some of you may be like, man, he's getting heated. I am getting heated because the science is there. The vast majority of products that we use in our life today are genetically altered in some capacity. Corn is genetically altered, albeit it took 10,000 years for us to alter it, but it is genetically altered. The raspberries, you go to the grocery store, you can find raspberries, strawberries, blueberries. You ever notice how massive they are? Why are they that massive? Because there's something called polyploidy. What does that mean? Plants have a remarkable ability to basically their fruit, the cells of their fruit can contain up to 16 times the number of chromosomes as it normally would. And in a eukaryotic cell, or like humans, which are made up of eukaryotic cells, that would normally kill us. But plants, it doesn't kill them. It makes the fruit bigger. And so how do we get the massive strawberries we see in the grocery store? Well, they're polyploidy. And so we, through selective breeding or direct genetic altercation, we select for the strawberries that are bigger, i.e. polyploid, and the bigger the strawberries we get from one plant, we breed that plant with another. And we just keep going until we get massive strawberries. Now, of course, there's a limit to how big they can get, but you'll never find a strawberry as big as you can find in the grocery store in nature. You never will, because that's a product of human intervention. But do we not eat it? No. And the brands that say non-GMO, they're still genetically modified. It's just we didn't genetically modify it with a plasmid, CRISPR, or a viral vector. So ultimately, ladies and gentlemen, before I end today's episode, what I'd like to do is I'd like to recap everything. So to recap, the vast majority of the science out there says that there are no directly observed negative health effects with the consumption of genetically modified foods, that, of course, with the caveat that they're only looking at ones that have been approved for human consumption. Number two is, in fact, the vast majority of products that we consume on a daily basis in our lives are genetically altered. Now, that may be through selective breeding or polyploidy or direct genetic modification using CRISPR or viral vectors. The reality is the vast majority of foods and items we consume and eat on a daily basis in our lives, you can't find in nature because they have been altered in some capacity, either over the past 5 years through direct genetic mechanisms or over the past 10,000 years through selective breeding. And number three is, ladies and gentlemen, stand up and question everything. And I know I say that a lot. I know that's my slogan, right? Stand up and question everything. But I think that especially reigns true here. Always question what you hear from people, even what you're hearing from me. I try and be as scientifically accurate as I can, but I encourage all of you to go out and do your own research, because it is that drive that not only furthers ourselves, but it furthers science and innovation. It's the drive to say, I'm going to question that. I'm going to stand up and question everything. I don't believe you. I'm going to do my own research. That drives science. And science is all about asking questions and testing our questions. So, ladies and gentlemen, I hope you enjoyed today's episode. I hope I made you at least a little more comfortable about GMOs, because the reality is there's nothing to worry about. Until a paper comes out that says, Atticus, you're 100% wrong, this disease and this disease and this disease has been linked with this particular genetically modified organism, there's no literature that suggests that any of the negative claims about it in terms of health are valid. Albeit, though, there are a lot of predatory companies like Monsanto that produce genetically modified corn and take advantage of farmers. That's an entirely different story. That's a story of politics and of law and of predatory marketing, not of the actual science itself. So, ladies and gentlemen, thank you very much for listening to today's episode of The Science Behind That. I hope you guys have a fantastic week. I'll see you all on Friday. And remember, as always, stand up and question everything.